When we think of evolution, we usually picture this super slow, gradual process, right? But bacteria, well, they play by a totally different set of rules. They can develop these incredible new abilities that leave our best medical defenses just scrambling to keep up. So, how on earth do they do it? Let's dive into the hidden world of bacterial superpowers. Seriously, think about it. For most species, evolution takes thousands, sometimes millions of years. But with bacteria, it's a whole different ballgame. One day, an entire colony is totally vulnerable to an antibiotic, and then, seemingly the next day, they're completely resistant. It feels like magic. So how do they pull off this incredible trick? This quote from our source material absolutely nails it. It really is like someone hit the fast forward button. Bacteria have found these ingenious and, frankly, sometimes alarming shortcuts to share survival secrets and just rewrite their own genetic rulebooks in real time. So here's our game plan. We're going to look at the genetic shortcut that sets bacteria apart. We'll break down the 4 main ways they share their secrets, check out the tools that make it all possible, and then connect everything back to the very real, very urgent challenge of superbugs. Okay, so to really get what makes bacterial evolution so special, we've got to compare the 2 main strategies they have for survival. The huge difference here really just comes down to speed. Mutation, that's the classic slow burn. It's like a random typo in the DNA that might lead to something useful over generations and generations. But horizontal gene transfer, oh, that is a total game changer. It's not a tiny edit. It's like instantly downloading a fully functional software update from a neighbor, a prepackaged toolkit for survival. This is their ultimate evolutionary shortcut. So, how does this high-speed data download actually work? Well, bacteria use 4 main methods to share their genetic secrets. Let's break them down one by one. First up, we've got conjugation. Now, people often call it bacterial mating, which is a fun way to think about it, but it's really all about strategic survival. This is the most direct method they have, where one bacterium literally reaches out, builds a bridge to another, and hand delivers a genetic package. And the process is just incredibly active. A donor cell shoots out this thing called a pilus, think of it like a grappling hook, to grab a recipient, pull it close, and form a physical bridge. As it sends a copy of its plasma DNA across, it keeps a copy for itself. And here is the absolute key. The recipient doesn't just get the new trait, it becomes a second donor. This just exponentially ramps up the speed at which a new skill can spread through a whole population. Okay, our second method is called transduction. This one is a little more indirect because it involves a middleman, a virus. To be specific, a type of virus called a bacteriophage, which has evolved to only infect bacteria. This analogy is just perfect. Imagine a virus hijacking a bacterium to make more copies of itself. But sometimes, during the assembly process, it messes up. It accidentally stuffs a piece of the host's bacterium's DNA into a new virus shell. So when that faulty virus goes to infect a new cell, it doesn't deliver a viral payload. It delivers a genetic upgrade from its last victim. And the consequences of this are just profound. Sometimes the gene transfer is totally random, but other times it's very specific. And here's where it gets really alarming. Through a process called lysogenic conversion, the virus itself can be carrying the weapon. The genes for the deadly toxins that cause things like diphtheria and botulism, they're delivered this way. A harmless bacterium gets infected, and the phage's own DNA transforms it into a dangerous pathogen. Our third mechanism is transformation. This happens when bacteria die and their cells burst open, spilling their DNA out into the environment. Other bacteria, if they're in a special receptive state called competence, can literally just scavenge these naked DNA fragments, pull them inside, and start using them. It's like finding useful scrolls of information that have washed up on the shore from a shipwreck. And what's really fascinating is that this very process led to one of the biggest discoveries in all of biology. Griffith's famous 1928 experiment showed this mysterious transforming principle was at work. It wasn't until 1944 that Avery, McLeod, and McCarty finally proved that this principle was, in fact, DNA. This was how we first confirmed that DNA is the blueprint of life. And that brings us to our fourth and final method, transposition. Now, this one's a little different because it's an internal affair. We're talking about these special segments of DNA called transposons, or jumping genes, that can literally cut themselves out and move to a new spot within a single bacterium's genetic library. Yeah, this is a great way to think about it. These jumping genes actually come with their own enzyme tools that let them cut themselves out and paste themselves into a new location. They can jump from the main chromosome to a mobile plasmid or from one plasmid to another, so they are constantly reshuffling the genetic deck from the inside, often moving really important genes, like for antibiotic resistance, to places where they can be more easily shared. So, we've seen the delivery methods, right? Now let's take a look at the actual genetic cargo being delivered, and crucially, how it becomes a permanent upgrade for the cell. Okay, at the absolute heart of this whole genetic sharing network are plasmids. These are small, circular rings of DNA that live separately from the main chromosome. Think of them like genetic flash drives. They replicate on their own, and they're the mobile vehicles for all sorts of powerful genes. They're optional, sure, but they are incredibly useful accessories. And the upgrades they carry are absolute game changers. Plasmids can carry the code for everything from antibiotic resistance and disease-causing toxins to the very machinery needed for conjugation itself. They are basically the currency of bacterial survival, and a single transfer can turn a totally benign organism into a major threat. But here's the thing. For a new gene to be useful in the long run, it has to be integrated. It has to become a permanent part of the blueprint. That's where recombination comes in. It's the molecular process that literally splices that foreign DNA into the host's main genome. Without this final step, that downloaded software update is just a temporary file, lost as soon as the cell divides. Recombination is what hits the save button. Okay, so we've journeyed deep into the tiny, intricate mechanics of bacterial genetics. Now, let's zoom back out and connect all of this to the real world and understand why this constant, high-speed networking has such a massive impact on all of our lives. Here's the bottom line. These gene transfer mechanisms, they are the primary engines driving our biggest threats from bacteria. They're directly responsible for the terrifyingly rapid spread of antibiotic resistance and for passing around the virulence genes that can turn a manageable infection into a deadly one. And when you put it all together, you really see the scale of the problem we're up against. Bacteria inside a sick patient aren't just isolated islands. No, they're an active, microscopic intelligence network, constantly sharing defensive toolkits using these plasmid flash drives. This rapid-fire networking is exactly what creates multi-drug-resistant superbugs, turning what should be a treatable infection into a life-threatening crisis. So we really need to get away from this idea of bacteria as just passive little victims of evolution, just waiting around for lucky random mutation. That's not the story at all. They are sophisticated genetic engineers actively and constantly shaping their own destinies with a speed and an efficiency that we are still struggling to keep up with. And this really leaves us with a final big thought to chew on. Bacteria have been running the most successful, most resilient, and most adaptive information sharing network on this planet for billions of years. So as we grapple with our own challenges in technology, in medicine, and in building resilient systems, what lessons can we learn from the original masters of the genetic network? The secrets to rapid adaptation might just be thriving right under our noses and under the microscope.